Imagine a beautiful place in a pine forest between two huge rivers. It's cold outside, about 5 degrees Celsius, and we see a cave in the distance. We can also see a big bear and a couple of deer hanging out by the river. And who else? There's a small group of people, perhaps a couple and their children. They're sitting by a fire roasting an animal. The woman seems to be preparing a piece of meat. Meanwhile, the children are sitting at a distance making stone tools for their parents. Let's think about this very detailed picture. Is this what a random day in the Paleolithic was like? And how do we know this? There are many different archaeological disciplines that work very hard to approach these remote time periods. Each of these disciplines contributes a little bit to the big puzzle of prehistory. Archaeological clues can be found in bones, stones, sediment, and other materials with high preservation potential. And it is the results of studying these materials that eventually become pieces of the big puzzle of prehistory. So why don't we take a closer look at one of these pieces? Let's take, for example, a bit of sediment that has been studied using the technique of soil micromorphology. Soil micromorphology is a branch of geoarchaeology that focuses on the microscopic investigation of undisturbed blocks of archaeological soil or sediment in search of clues about human behavior and past environments. Let's take a look at the kinds of information that we can obtain from this technique. By using microscopes to zoom into archaeological sediment at very high magnifications, we can see things that would be invisible to the naked eye. And not surprisingly, things are not always what they seem. We can see, for example, if the deposit is intact or if it has been disturbed throughout the years. It is very important to know the degree of integrity of an archaeological deposit before we interpret it, because it could have been moved to a different location, it could have been mixed with older materials, or, even worse, large parts of it could be missing. We can also use the technique of soil micromorphology to find microscopic clues about past climate and past vegetation. We can also take a look at the kinds of activity that humans were doing at a certain place and the kinds of tools they were using. Also, we can find food residues. We can also see if humans occupied a place for a long time or only briefly. In other words, imagine a cave site, a very famous Paleolithic cave site, in which prestigious scientists go and find very old DNA of humans. They also find some stone tools made by humans and some bones that humans ate. However, are they all associated? Are they all from the same time? They're all inside the same soil. But a closer look in the microscope maybe tells us that the stone tool is 4,000 years, 5,000 years older than the bone. And what else? The DNA dissolved in the water in a much more recent time and found its way next to the bone. So imagine if we did not have the opportunity to know all these things. These little details can be learned by looking at the fine microstructure of the soil. Under the microscope, we can really tell if different associations of tools and bones, even DNA and other residues, belong together or not. The technique of soil micromorphology has been around since the 70s, more or less. It has been very important in Paleolithic archaeology thanks to Paul Goldberg, who is the pioneer. These old archaeological contexts can be quite poor and monotonous, without any visible organic remains. It's a time before architecture, and often we are only left with stone tools and butchered bones. It is difficult to get a picture of human life with such little evidence, and also to know if the few remains all belong together, and even if they're in their original spots or not. Besides the Paleolithic, the technique has also been applied to recent prehistory, and even Roman, Greek, and medieval sites. Less work has been done in Bronze and Iron Age sites, but this is quickly changing with the new generation of scientists. Let's take a look at how the technique works. The main procedure is to make thin sections from archaeological sediment and observe them using a petrographic microscope. First, blocks of sediment are collected from the context of interest at the site. Many times these have to be cast in gypsum to preserve their integrity, especially when sediments are very loose, like ash from a fire.
Hey, how should I label the block? On one face only? And is it important to mark where the backside is?
Yes, that's very important. And also remember to use a black permanent because other colors tend to fade away with time. So here, first you have to write arrows pointing at the top of the block in every face. And then write a target symbol at the top. This is crucial because if we don't know which side is up, we lose the time dimension. Down is older and up is younger, and this is very important. Also, if the block comes from a profile, which is not our case, you want to indicate which side comes from deeper inside, which side is more fresh. Because normally the exposed profile gets reworked by insects and roots and even animals, and you don't want to work with that part, okay? And finally, remember to write the ID of the sample in every face so that it doesn't get erased in the lab. And that's it. Now you're all set.
Okay.
So what do we study once we are looking in the microscope? One of the things we study is the microstructure. Whether the sediment is loose or compact, if it has cracks, if it is crumbly, if it's platy. Each of these features are informative of processes that occurred in the past, natural processes or human processes. And therefore, they give us clues about the lives of humans that lived there when the sediment was being deposited. For example, an archaeological sediment with horizontal lenses usually indicates a climate with seasonal freezing and thawing, or a very compact sediment with many little charcoal bits and tiny bones broken in place usually indicates that the place was trampled and that a human living floor was right there. At the lab, the blocks are dried in the oven at a low temperature for 1 or 2 days, and this makes sure that all the moisture in the sediment goes away and that the pores are empty to make room for a mix of polyester resin that penetrates the block. The idea is to plastify the block, to make a solid block of sediment that can be cut very thinly to make thin sections of it. We always take into account the orientation of the block. If we lose the orientation, it could be disastrous. Let's remember that in an archaeological site, time is represented vertically. Upwards is more recent, downwards is older. So that our block has to be carefully marked with an arrow indicating upwards so that we don't lose the track of time. Once the block is plastified, it can be cut into approximately 1 centimeter thick slabs. And these slabs are glued to a glass slide. And then they are cut with a precision saw until they're about 1 millimeter thick. Afterwards, they go into a grinding machine, and this 1 millimeter thick film of soil stuck to a glass is ground down to only 30 microns of thickness. This process is all quite difficult and it takes great skill and craft. There are very few micromorphology thin section manufacturers out there, and they are very well appreciated. Finally, the finished product is a thin section that can be analyzed under the microscope. We use petrographic microscopes, ones with transmitted light from the bottom. The light goes up through the sample, and you see particular colors depending on what the particle is. The light goes up through the sample, and you see a variety of colors that are all specific to the material you're looking at, so that you can identify it by its color. There's different tricks in this. For example, cross-polarizing the light, just pointing all of the light beams into a single direction. The same as polarized sunglasses, with the same principle. To block some of the light and allow only a few parallel rays to go through. We also use different filters to identify different minerals. A common filter is the blue light filter. Blue light allows you to excite particles that contain some minerals called phosphates. And phosphates are what excrements and bones are made of. So identifying phosphates with blue light can give you clues about the presence of bone or an excrement from an animal. Let's take a look at some examples to understand this a little bit better. There's a famous case study. It's the Paleolithic site of Zhukotian in China. Here, micromorphology analysis revealed that what appeared to be burned sediment from a very old Paleolithic fire, the earliest evidence of human-made fire in the world, actually was just sediment that was reddened by oxidation from a puddle. There was a puddle at the entrance of the cave. And this puddle dried up, making the sediment turn red. It was not a fire. This was discovered thanks to micromorphology. In another case, at the Neolithic site of Asikli Huyuk in Turkey, microscopic excrement residues were found. And this indicated that there were stables there, goat or sheep stables. And they helped discover that animal domestication occurred in that region much earlier than expected.
So, how can we know that the features we're looking at are contemporary to the humans that live there? I mean, these channels are probably from roots, but they can be very recent roots, right?
Yes, exactly. Well, sometimes it's not possible to tell the difference, but we can look at some features. For example, if you look at features near the surface, like horizontal cracks that are caused by seasonal frost, and if these cracks appear next to archaeological remains, then probably we can say they are contemporaneous. But there are other features that don't behave the same. There are features that appear very deep in the profile, like for example clay coatings. The clay coatings, they form because of a water action. Water percolating the soil because of rain or irrigation affects the sediment. So if you find these clay coatings next to archaeological remains, probably these remains were actually underground before the clay coatings were formed. So we cannot say they are contemporaneous.
And how can we tell that the sediment has suffered diagenesis? I mean, we're looking at something that's really old, it's Paleolithic.
Yes, so we can have some insights if we check in the fine matrix while looking at the thin sections, for example, here. So the first thing we have to do is to increase the magnification. Here with the objective, we change it to 10x. And then we check with the cross-polarized light, with this, to check the fine matrix. And we see the clay here, and the birefringence is not very bright, so you see almost black, and that means it has been phosphatized. So in the beginning, probably this clay was very bright and calcitic. When we have this type of clay, it means that the clay has been decalcified and it has been phosphatized because of the action of organic matter and excrements. But if you have also this clay, you can check at bones, and you can see if you have also some diagenetic bones there. The bones usually have this gray, bright color of the collagen fibers, but if you have diagenetic bone, you will see it also isotropically, like very dark and very black almost.
As we have seen, the technique of soil micromorphology is a technique used in archaeology that has great potential. It has demonstrated through the years that using these slices of the past, we are able to reach out and observe the archaeological sediment at timescales that are so narrow, so close to human timescales.